All right, I see folks are joining, continuing to join, but we're right at 2 o'clock, so we're going to get started to make sure we have plenty of time for our presenters and everything. Hello, everyone. My name is Emily Knight. I am a manager with the Lenfest Ocean Program. For those of you who do not know us, the Lenfest Ocean Program is a grant-making program that funds ocean and coastal research projects to address the needs of decision makers and stakeholders. And you can learn more about us and our projects at lenfestocean.org. And while you're there, sign up for our newsletter. And if you're on Twitter, be sure to follow us at LenfestOcean, and we'll be live tweeting this webinar on the hashtag LOPwebinar. So feel free to engage with us there. This is actually the third webinar in a monthly series that Lenfest has been hosting, highlighting our projects from the Climate Resilient Fisheries Grant Portfolio. This is a collection of projects that the portfolio started in 2018, so many of the projects have been ongoing for several years. And we wanted to, throughout 2024, highlight the results coming out of each project. So we are supporting a number of projects where the scientists are working with managers and stakeholders all over the US and abroad to better understand how climate change will impact fisheries and help fisheries managers and communities develop solutions. So we have a range of topics in this portfolio, from shifting fish stocks, understanding climate vulnerability of communities, to more ecosystem related questions like this project, and providing guidance on adaptive management for fisheries. So my colleague, Kayla Ripple, is going to drop links in the chat. She will drop the landing page for this collection of projects so you can learn more. And I will also say the next webinar in this series will be Wednesday, April 24th at 2 p.m. Eastern, 11 a.m. Pacific, and it will be on using predator-prey relationships to predict shifts in marine species distribution. And Kayla will drop the registration link for that into the chat. But to today, we have joining us Tessa Hill from University of California, Davis, as well as Esther Kennedy from the University of California, Davis as well. In this project, Tessa and her team of researchers analyzed geospatial patterns of ocean acidification and other environmental stressors like temperature and oxygen to identify where and when organisms along the West Coast might be vulnerable. And they focused on culturally and commercially valuable species like clams, crabs, oysters, and abalone. And Kayla will drop into the chat the project page, as well as the papers they have had out from this project. And I will also say, you may have heard that Tessa and science writer Eric Simon have also come out with a new book, At Every Depth, Our Growing Knowledge of Our Changing Oceans. And Tessa will say a few words about the book at the end, but Kayla will also drop a link to that so you can get your own copy about this and other really important issues about climate change impacts on the oceans. I will let Esther and Tessa get into the details, but this project takes on a really complex topic. Ocean acidification has had big impacts on the West Coast, especially in terms of the shellfish industry. But one thing scientists have often recognized was the need to study OA in the context of a changing ocean. It's not just pH, it's warming temperature, it's changing oxygen, and all of those things are interacting over space and time. So this project takes on a really challenging question, but a really, really important one. And we've been thrilled with the progress they have made and the papers they have produced in this. In fact, Esther will be finishing her PhD on this coming summer. So keep an eye out for that. And she's done such great work with this. So reach out to her. So before we get started, just a few logistical things. To prevent any feedback, all attendees are muted. Please feel free to drop your comments or other resources into the chat box during the webinar. For questions, which I will read to the researchers at the end once they're done presenting, if you can, please submit them into the Q&A box. That just makes it easier for us to manage the queue. We will try to keep track of the chat as well if you happen to put questions in there, and do our best to get to those. If we do not get to your question, please feel free to reach out to us or the research team after the webinar. If we run out of time for questions, we will actually stop this webinar a couple minutes ahead of the hour. But you can always follow up. We will download all of the questions, and we're always happy to follow up. So lastly, the webinar is being recorded, and we will distribute the link broadly afterwards. So please share this with others, and again, follow up with the research team. So otherwise, I will actually turn it over to Tessa.
Thank you so much, Emily, and thank you all for being here today. Let me share screen, and then Emily or someone, can you just confirm for me that that worked?
It looks great.
Okay, lovely. Thanks so much for being here. Esther and I are very excited to share with you some of the results of this project that has been going on for several years. I have the pleasure of providing a little bit of context, how we got into this project, a little bit of background information, although I suspect many of you know quite a bit about this subject. I'm going to hand off to Esther, who's going to then tell you sort of a series of vignettes about what we learned, highlighting a few papers that have come out, as well as some papers that are currently still in progress. And then she'll hand back to me to sort of wrap that up, and we'll end with some sort of connections to other work that's ongoing. So the context for today's talk really centers around the many different ways that the ocean is experiencing change today. And I love this figure from the California Fourth Climate Assessment, because it really shows many different ways that we have to think about a changing ocean. Today's talk is really going to focus on what's in that lower left orange box. So we're really thinking about how ocean chemistry interacts with a changing ocean, specifically ocean acidification and hypoxia, alongside temperature change, and sort of made a decision here to not provide a primer on ocean acidification and hypoxia, because I think many of you who registered for the webinar probably know quite a bit about that. But of course, Esther and I would be happy to, explain things if we've moved too fast over these items. But I did want to sort of hone in on the fact that of all these many different things that are changing in the ocean, what we're going to talk about today is trying to get a handle on these sort of three overlapping stressors: temperature, hypoxia, or changes in oxygenation, and ocean acidification, or fundamental chemical changes to the ocean. Our research group, which I'm going to introduce you to in just a minute, has been interested in these ideas for over a decade. At UC Davis Bodega Marine Lab, we've been doing work on ocean acidification for somewhere between 15 or 16 years, somewhere in there. And so the jumping off point for the project we're going to tell you about today, of course, comes from this long-term interest in trying to understand the implications of ocean chemistry change. And so, one approach to that over the past 2 decades is that through laboratory and field experiments, we've tried to understand the impact of ocean acidification on widely distributed species. As Emily described, these species are often commercially important, ecologically important, or culturally important to the West Coast of the US. And we've learned quite a bit about a myriad of different responses to the stressor of ocean acidification, particularly with these sort of marine invertebrates. Taking a broader view, thinking about food webs and carbon cycling, and this time I'm highlighting work not just of our own group, but of others as well. What we know is that ocean acidification has the potential to negatively impact species that really underpin food webs in marine systems, and also our understanding of carbon export and carbon cycling. So we know that there are very significant impacts on commercially, culturally, economically, ecologically important species. We know that there's the potential for these chemical changes in the ocean to really shake up, everything from predator-prey interactions to food webs to carbon cycling. And that knowledge was a jumping off point for what we want to talk to you about today. And the other jumping off point were some hints about how variable this West Coast system can be. And what I'm showing here on the left is a compilation of data where 3 research groups got together using NOAA PMEL shipboard data led by Richard Feely, NSF Omega's data led by Francis Chan, and some shore-based sites that we were monitoring, about 50 sites along the US West Coast in our group. And our 3 research groups got together and wrote a paper in 2016 that showed what happened when you merge those 3 different data sets. And part of what we see is this very powerful and interesting mosaic of pH along the coast. And so what you're looking at is surface ocean pH in those colors, where the sort of yellows to reds are very low pH, more acidic conditions, and the blues are more buffered conditions. And we see a tremendous amount of variability in this system. And that variability is very scientifically interesting. It creates sort of a natural laboratory where we can test ideas about adaptation to future change. But it also struck us that variability opens a window. It opens a window for us to think about local scale management, local scale adaptation and solutions that we can think about as a region or as states, Washington, Oregon, and California in this case, in terms of how we manage this system, because the way that ocean change is going to proceed in the system will not be uniform. It's not even uniform as a starting point. So that was, I think this was a point of excitement. And we thought, well, this is just 3 data sets put together. What would happen if we try to pull together many, many more? And that's what Esther is going to get to talk to you about in just a minute. So our 2 sort of overarching questions that we're going to cover today are: what are the geospatial patterns of acidification, oxygen, and temperature for the US West Coast? And where are key species vulnerable to those overlapping environmental stressors? This work has been generously supported by the Landfest Ocean Program. We also received some support from the California Ocean Protection Council to answer some California-specific questions. And as I will get to tell you at the end of the talk, this work also integrates with a broader project thinking about social science implications that has been supported by the NOAA Ocean Acidification Program. And we're not actually going to get to present on those social science aspects today, but I am, at the end of this talk, going to tell you about how that integrates with these 2 primary questions that we're discussing today. I mentioned that we're here on behalf of a big research team, and so I want to acknowledge everyone on this screen. We're presenting information today from investigators at multiple different universities, as well as collaborations with the California Ocean Science Trust. And all of these team members have been essential to the success of this project. And Esther and I have the real pleasure today to be able to highlight some of these individuals and the way that they've taken a leadership role and made some really important discoveries. So you're going to hear and see us highlight several people's work as we share those vignettes with you today. Before I hand off to Esther, I'm almost handing off, but I did want to tell you that one of the things we really appreciated about the Landfest Ocean Program model of doing this work is that it was expected that we engage early and regularly with people who would use this information. And in fact, we engaged with end users in the proposal writing stage of this project. And that was a starting point. It then was fairly continuous from there. So I just wanted to give you a sense at the start of this talk of what that engagement looked like. And then at the end, I'm going to tell you about some of the outcomes that were derived from that engagement. LenFest helped us host a public webinar at the very beginning of this project. So kind of like this one, but in the very early stages. We then hosted an in-person workshop with managers from California, Oregon, and Washington, as well as other end users present, again, at the beginning of the project. And you'll hear us reference throughout the talk today that discussions in those early webinars and workshops actually shaped the directions we took with this work. We maintained some of those relationships, many of those relationships through sort of individual meetings and check-ins throughout the project. And we also held a series of office hours in 2022, sort of modeled after the professor idea of office hours. They were drop-in. We had a series of these where we would provide updates on the project, take questions, take suggestions, etc. I'll also mention, I sort of hinted at this on a previous slide, there was also a California Ocean Protection Council project nested within this, and a NOAA ocean acidification project that was moving alongside this. And both those projects explicitly had advisory boards. And so while the LenFest project did not have an advisory board, this project benefited from interactions with those two advisory boards, which is definitely a model I would recommend. I think it really helped us to have pretty regular check-ins with folks who were going to use this information. So the sort of the final thing I will mention, one of the things we learned early on in those workshops actually was the importance of being clear about what we could and could not do in this project. So I'm going to mirror that here in the webinar today and do some level setting around what we hope to do, what we were able to accomplish, and things we knew that we couldn't do. So we did not intend for a strong focus on estuaries, including Puget Sound. There was a very strong interest in Puget Sound when we started this project, and we generally felt like there is so much expertise in that, and it's, you know, that expertise is not necessarily our research group's expertise. And so we were pretty clear from the beginning that we were prioritizing open coast data sets, and that's what you're going to see predominantly today. We do have some data from estuaries along the US West Coast, but it's much more sparse than the data we were able to pull together from the open coast. We did not set out to create predictive oceanographic models. So one of the things you're going to see today is that we are looking at measured conditions over the past many decades. It's not a predictive analysis. It's actually what the conditions have been very recently or in the past. However, this work could be used to strengthen predictive models. And of course, we would love to work with other investigators on that. And we did not incorporate privately held data sets. So the whole idea behind this project is that we focused on publicly available data that was already out there on data portals or publications. And our goal was to synthesize that so it was all in one place. And so we didn't do a lot of data digging. If the data were available, they were incorporated, and if not, they were not. So with that, I'm going to hand off to Esther, and rather than telling you what we didn't get to do, Esther has the fun job of telling you what we did get to do.
Well, thank you very much, Tessa, and thank you, Emily and Vicky and Kayla for having us. Yeah, so I guess I do get to talk about what we did do. But just before that, you might be wondering, don't we already have a bunch of data compilations? Why did they do another one? So a couple of these compilations include CODAP-NA, which is a compilation of bottle samples from oceanographic cruises. Senkus has an excellent data exploration tool that they maintain. And these two particular compilations represent kind of two different philosophies that we were trying to bridge. So CODAP-NA is a highly restrictive product. It doesn't include anything that is from shore or from autonomous sensors, but it has extremely high data quality and extremely high, everything is very consistent. Whereas the Senku's data portal is very permissive, but the flip side is that data are not standardized, they're not quality controlled, and they are very difficult to sort of work with because they are all in different formats. So we felt there was a need for a dedicated coastal product that was specifically set up to optimize warming, deoxygenation, and acidification studies, and that spanned from sort of the near shore to the open ocean, but that we set up so that it would sort of be high quality and easy to work with. You can go ahead to the next slide. Thank you. So of course, the synthesis effort was made much more possible because of the previous inventory work of many others. In particular, the California Ocean Protection Council and the IUCE network have put together some great inventories on the West Coast, and those were invaluable to us as we tried to find data sets and pull in data sets from throughout the West Coast. So thank you both. Our synthesis is called MOCA, the Multi-Stressor Observations of Coastal Hypoxia and Acidification. And I will say I'm overly proud of that acronym, but if you do not like it, please send me comments. So in the maps on the left, you can see the coordinates of all of our incorporated temperature, dissolved oxygen, and pH observations that this compilation includes. And so you can see that we kind of focused on nearshore data sets, but some of these records extend very far offshore. And you might notice that none of the pH records extend very far offshore. And this will be a bit of a theme in this part of the presentation, that our ocean acidification monitoring lags a bit behind temperature and dissolved oxygen monitoring in both space and time. So this compilation includes moorings, it includes research crews, bottle and cast or CTD observations, and shore-based samples, and they're all combined into a common format. They're publicly available for download at NCEI. You can also, we have code examples up as well if you want to work with the data. And in addition to the parameters I've shown here, it includes the full suite of the carbonate system, as well as nutrients and chlorophyll when they were available. But there are a couple of obvious data streams it does not currently include. So those are drifters, gliders, percolators, a few I'm sure we missed, some publicly available data sets. And I will note that we're planning to push a substantial update to bring the data up through 2022. And that will happen as soon as I do it, which hopefully is soon. You can go to the next slide. Okay, so just a little bit of information about like when we pulled all this data together and at its most surface level, what does this data tell us? Most specifically, it tells us where and what and when are we monitoring. I'll show a few of our research projects that build off of this as well. But first, yeah, just a couple of these maps of like, when are we actually monitoring what in the ocean? So here on the left, you're seeing a kind of map through time. The y-axis is latitude. It extends from the Southern California border to Canada. Just imagine a line going up the coast. And the x-axis is time from 1985 to 2020. And the color is the density of our nearshore surface observations. So data within 50 kilometers of shore and 25 meters of the surface. And as you saw in the previous slide, ocean acidification monitoring, so any carbonate system monitoring, is not present anywhere before 2007 and is just much less spatially extensive than temperature and dissolved oxygen. Secondly, you'll note that dissolved oxygen and OA monitoring are sparser in Northern California and Oregon and Washington, but we know from previous research and sort of ongoing extreme events, that these areas are particularly liable to have hypoxic and acidic events. So you might argue that these are the areas where we need monitoring most. And lastly, one interesting takeaway from this was that the peak of our spatial coverage was from 2010 to 2015, and we have actually lost some spatial coverage since then, except in Southern California. Can go ahead. Another way of looking at gaps in this observational record is to look at how well we're monitoring throughout the year. So the dotted line in these graphs on the right shows an even split between the 12 months of the year. You can see in the top graph that temperature, salinity, and dissolved oxygen are slightly biased towards summer months, but not too bad. And really only March and December are poorly sampled. But in the bottom graph, where we have ocean acidification parameters, pH, total alkalinity, and dissolved inorganic carbon, or DIC, there's a lot more concentration in May, August, and September. In some ways, this reflects, in a lot of ways actually, this reflects the ease of taking different types of measurements. So things like temperature, salinity, dissolved oxygen, and pH to some degree can be pretty easily monitored with autonomous sensors that can stay out all year. But to get really high quality ocean acidification observations, you often need to monitor total alkalinity or DIC, and those typically need to be individually collected, often on a ship, preserved and analyzed in a lab. And so we only have samples when sort of the ships are running. So that is just one of the many ways that our observational record can be like a little bit harder to work with in some ways than a model, because we do have these kind of biases built in. So finally, we can take a quick look at the seasonal and regional distribution of our nearshore data, which again highlights some gaps, as well as some interesting relationships between potential temperature, oxygen, and pH stressors. For a quick box plot primer, if you're unfamiliar, all of the dots are observations of, this is all of our nearshore data within the 100-meter sort of bathymetric contour. And the box is marking the middle 75% of the data, with the bar in the middle showing you the median. Okay, so first you can see some of the seasonal cycle you would expect. It does get warmer in the summer and it is colder in the winter, for example. But you can also see, again, how sparse our dissolved oxygen and pH records are in some regions, particularly Washington for dissolved oxygen, and Washington and Oregon for winter pH. And finally, you might note here that the warmest months are those with the lowest median dissolved oxygen and pH, rather than necessarily the months you might associate with peak upwelling, which is sort of more April to June. Upwelling, of course, is still common through the summer and alternates with periods of relaxation that can be very, very warm. So spring and summer are where we're going to see the most variability and the most potential for extreme conditions on both the high and low ends. Okay, so now we can transition a little bit into what all this data actually means. What does it tell us about impacts? Go ahead, Tessa. Great. So one of our lab's previous postdocs, Dr. Sarah Hamilton, used the MoCA compilation to ask how multiple stressors were impacting California marine protected areas. So she used daily averages of all surface observations within 10 kilometers of a given MPA and excluded MPAs that did not have at least 6 months of continuous simultaneous monitoring of temperature, dissolved oxygen, and pH. Of all the MPAs in California, which I will note, there are 124 of them, they're shown on this map in the red polygons, only the 9 bright circles on this map met that criteria. So only 9 MPAs had 6 months of monitoring of temperature, dissolved oxygen, and pH. An additional 10 MPAs, so 19 total MPAs, had sufficient data to examine temperature and dissolved oxygen alone without pH. You can read more about those in her paper, which is published in ICES. So to define stressful conditions, Sarah used dissolved oxygen and pH thresholds that are identified in the literature as harmful to many organisms. These thresholds were 100 micromoles per kilogram for dissolved oxygen. That's the vertical red dashed line on all of these plots. And a pH of 7.75, which is the horizontal dashed line on all of these plots. Temperature stress is a little less clear-cut. Temperature stress thresholds are less conserved across taxa. They're more variable across geographic regions. So for this, Sarah calculated the temperature anomaly for a given location and time. So warm colors on these plots are showing temperatures that are warmer than usual for that location and time, and cooler colors are showing colder than usual. So each of these 9 plots shows the surface pH, dissolved oxygen, and temperature observations from within 10 kilometers of that MPA. And I know the map is really small here, but note that the map label colors are corresponding to northern, central, and southern California. So you don't really have to match them too much, just know it's sort of north, middle, south. There are a couple clear takeaways from Sarah's work. So first, MPAs with strong, nearly linear relationships between dissolved oxygen and pH are really strongly influenced by upwelling. You'll note that the lowest oxygen and pH the lowest dissolved oxygen and pH values in these MPAs are associated with very cold water, as that sort of deeper water that is cold, acidic, and hypoxic gets pulled up to the surface. Upwelling, of course, is a feature of the California current, but you'll also note that these upwelling events tend to cross pH, stressful pH or dissolved oxygen thresholds, or both. Secondly, in the parks that are not as influenced by upwelling, you'll note that these stressors occur much more in isolation from each other. So you'll have either low pH, low dissolved oxygen, or high temperatures, but not usually any combination of those at the same time. Most MPAs are crossing both dissolved oxygen and pH critical thresholds, although the ones 3 out of 4 in Southern California are less likely to. And lastly, Sarah was able to calculate the percentage of time that each of these MPAs sort of spends across these critical thresholds, and that was highly variable. It ranged from 0 to 75% of the time for individual stressors, and up to 25% of the time for multiple stressors. Okay. So to present another quick vignette from another one of our wonderful postdocs, Dr. Rachel Carlson is investigating coastal microclimates very much on the scale of sort of human communities, so very, very small spatial scales and very small management zones. And one big part of her work is investigating the influence of fresh water on local carbonate chemistry. So here in this figure, you can see streamflow discharge rates from the USGS, but along the coast, you're seeing the location in red of all of our pH observations in the MoCA dataset within 10 kilometers of the coast. So she's looking to tie the variability in freshwater influence to sort of pH microclimates along the coast. And a big hope with this work is that by elucidating the impact of freshwater on these microclimates, that gives policymakers a lot more levers to affect their sort of local environment. Freshwater is more tractable to being managed and mitigated at the local or state level. And so as a local policymaker, you might be able to have a lot of influence on sort of what's coming out of your stream, but you only have so much ability to affect ocean acidification writ large. So, and sorry, just got lost for a second here. But so moving on with Sarah's work, or sorry, Dr. Rachel Carlson's work, moving on with Rachel's work, part of this is about streamflow data, or as I mentioned, part of this is with streamflow data and how streams are impacting the local microclimate. So showing some of her preliminary research, she's looking specifically not just at discharge or stream flow, but also soil pH and soil alkalinity to see how those impact coastal pH and coastal alkalinity. And so this is very new. This is a very new project. This is very new work. And some of her preliminary results here are showing our ocean alkalinity, or sort of the buffering capacity of the ocean, our ocean alkalinity observations from near USGS, I believe it's USGS soil pH measurements. And you can see that there's a lot of variability here between soil pH and the sort of associated ocean alkalinity, but there is a bleeding lower edge to this graph in where lower soil pHs can be can sort of pull the ocean alkalinity much further down. Sarah is also doing some very interesting work on mussel calcification using these observations from the MoCA data set and streamflow to calculate the effects on mussel calcification. And so at the end of this, she will have quite a bit of information about coastal microclimates, what policy levers managers might pull to mitigate those effects, and then what the impacts might be on coastal calcifiers. All right, so to kind of wrap up the sort of MoCA side of things, this compilation has provided a really excellent resource for investigating deeper questions about risks and consequences of warming and deoxygenation and acidification. Despite the fact I have maybe belabored the gaps in our observational record a little bit, but despite the gaps in our observational record, this is a really excellent resource, and we really have been able to quantify some of these multiple stressors that we're seeing in the ocean and where and when those stressors are coming to the fore. But there's a really big piece missing from what we've presented so far, which is how do we connect these stressful conditions to the organisms that are actually getting stressed? How do we actually move this to local ecology? So as Tessa had already mentioned, as the second question in this research, we're interested in investigating where and when key coastal species are exposed to one or more of these environmental stresses, and then better identifying what the critical oceanographic thresholds for those species are in the first place. So we've learned a lot in the last 15 years about vulnerability of marine species to environmental change, but there's still a lot we don't know. And I recognize you probably can't read this figure, but here are the things you need to know about it. There's a list of species on the left-hand side, and there are many species here. And then all of the things highlighted in red are things we don't know, which is to say there are a lot of things we don't know about a lot of species. So one aspect of our project was to investigate the vulnerability of species of management concern identified, say, on this graph. And we'll present two quick projects related to that. So first, my lab mate Megan Zulian's work, which characterizes how frequently critical species encounter stressful conditions and how well we even know whether they are encountering those conditions. And then secondly, Ben Walker in Christy Croker's lab at UC Santa Cruz has been working to refine our understanding of those stress thresholds. Okay, so one of the crucial underlying questions to species vulnerability is, are we even monitoring at the right places and at the right time for a given species' vulnerable life stage? So to address this challenge, Megan's work asks two related key questions. First, how much exposure do these 5 invertebrates pictured up top have to stressful conditions, given what we see in the observational record, and given their distribution at a given life stage? And secondly, How much confidence do we have in these exposure rates, given these observational gaps that I've talked about a lot? Several recent studies have examined species vulnerability and exposure using modeled oceanographic conditions. And Megan has followed these established methods, but she's coupling her exposure analysis to those observations in the MoCA synthesis. So first, Megan gathered distribution information, such as spatial and depth extent, on each of the invertebrates at each of their major life stages, so egg, larval stages, juvenile, and adult. She then used oceanographic observations from MoCA to calculate the frequency of pHs below a threshold of 7.65 or dissolved oxygen concentrations below 61 micromoles per kilogram to calculate an exposure score that she rated as low, medium, or high. She combined the exposure score with a consequence score based on the organism's response to acidity or hypoxia. So did the organism have essentially no response to crossing those thresholds? Are there sublethal responses, or are they dying? Separately, she also rated the uncertainty of these exposure scores based on the completeness of our observational record as it related to that specific species and life stage. Here you can see Megan's results plotted by uncertainty on the x-axis and vulnerability on the y-axis. And the grayscale here is corresponding to life stage. So younger life stages are white or light colored and adults are black. Overall, vulnerabilities are low. You can recall that the vulnerability score can range all the way up to 9. And most of those higher vulnerabilities, those species that are sort of edging up into moderate vulnerability, those high numbers are driven by very high consequences as opposed to very high exposures. So she's not finding very high exposures to stressful conditions, but some of these species are sort of vulnerable by dint of just being liable to, say, die if they cross any of these thresholds. But uncertainty, on the flip side, is often moderate or significantly higher than the vulnerability score, especially for select life stages and especially for pH. And this work is really underscoring the importance of tailoring our monitoring networks to our species of interest. We really can't predict species impacts without a strong understanding of what conditions they are actually experiencing. And so with that, I will hand it back to Tessa.
Okay, and I get to tell you a last vignette where I attempt to represent our amazing colleagues at UC Santa Cruz, Christy Croker and Ben Walker. So wrapping up this section, sort of thinking about species impacts and how they connect to the oceanographic synthesis. A major milestone we are hoping to achieve in this project is developing some species-specific maps that describe the impact of multiple stressors on key species for the West Coast. So for these species that may be commercially, economically, culturally, environmentally important, we might want to have a map that thinks about the cumulative impact of these multiple stressors. To do so, Ben and Christy have developed a meta-analysis of available experimental literature on response to ocean acidification and hypoxia to develop those cumulative indexes that we could map. The goal of this would be to combine those findings with our MoCA data set to develop a species-specific map for each species that would be of interest. However, one of the things we learned very early on in those workshops with managers, they brought to our attention that while we have significant knowledge about many of these species and taxa, there was a knowledge gap, a data gap that needed to be addressed. While we know quite a bit about some life stages of Dungeness, there was a data gap in our understanding the combined impact of pH and oxygen on juvenile Dungeness crab. So a key outcome of our research and a step towards this sort of combined synthesis of organismal impacts and the MoCA data set was to address this knowledge gap around Dungeness crab. And so Ben has been leading a series of experiments that I'm going to attempt to summarize for you here today. And I am using notes because I don't want to mess this up. So first, Ben and Christy raised juvenile Dungeness crabs in a static system, where a combination of pH and dissolved oxygen conditions were dialed in at set temperatures. You can see those conditions along the x-axis of the graph on the right. Growth of juvenile crabs was relatively stable across all experimental treatments until the final, most stressful treatment, which is on the far left side of that graph. The response of declining growth in the lowest pH, lowest DO, and coldest temperature treatment indicates a potential threshold that may be reached during current or future upwelling conditions that cause crab growth to decline. However, they wanted to add a layer of complexity to this analysis. So Ben and Christy also placed juvenile Dungeness crab under a variable system where pH and dissolved oxygen were oscillating, much like what you might expect during an upwelling and relaxation event. The frequency of that oscillation was also changed. So some experimental treatments had short duration upwelling and relaxation periods, and others had longer, 20 days long. Ben's preliminary results indicate that the duration of variability is key in understanding the impact of those stressors. The short duration events actually had a more negative consequence than the longer duration events. So with this information, our team is now working together to try to reach, what I just mentioned, a sort of a final milestone, thinking about pulling together that meta-analysis, that synthesis of organismal impacts, thinking in a cumulative impact space. So all the different stressors layered on top of each other in the context of what we can see in the MoCA data set. I did, before we finish, I did want to come back to how we've connected with communities, end users, and decision makers, so that we can think about how this work has been pulled into products and outcomes that people can use. And I just wanted to give you a few examples of ways that this has been accomplished. And I'll just highlight a few off this list. One is that we've been thrilled to partner with Senkus to get all of the data in the MoCA synthesis up on a data portal that is easier to use, more user-friendly than a data set that requires perhaps a programming language to navigate tens of millions of data points. So we've been really excited to work with Senkus on making sure that other people can actively use and plot data from this data set. We've been working closely with National Marine Sanctuaries sort of all along through this project, but I will also point out that that work that Sarah did on the California MPAs was explicitly asked for and supported by the Packard Foundation to have it come in time, in close timing with the MPA decadal assessment that was happening in California. So the idea of thinking about how we assess and evaluate and plan for future MPAs, having this knowledge of oceanographic data and vulnerability being integrated into that process. Similarly, the National Park Service asked us to develop a report on how these parameters are impacting national parks within California that have coastal real estate. So thinking about how the parks have potential to manage or mitigate some of these impacts. And I've listed off some other groups here that we've been in close collaboration with all along, close conversations where they're using these data for decisions that they are making. I hinted at the beginning that there was a social science aspect to this, so I'm gonna end with that point. I think all of us, many of us on this call know that our vulnerability to ocean acidification and hypoxia as stressors comes as some combination of oceanography plus the species that we are concerned about, and ultimately people, coastal communities that are reliant upon a healthy ocean. And as this paper led by Julie Ekstrom years ago summarized so well, we can think about both the exposure that the ecosystem is experiencing, which is in purple colors here, as well as the social vulnerability of the people in that system, in this case described by reliance on shellfish as an income source, and those are in the red colors. And so we can think about, ultimately when we think about vulnerability and adaptation, we're thinking about this integration, right, of the oceanographic system with people. And so with that in mind, I'll sort of hint to the fact that we've been integrating this work all along with that of social scientists to understand the adaptive capacity of people along the shore. We weren't really able to summarize that project in the length of this webinar, but our amazing social science team, led by Anna Spalding at Oregon State and Ariel Levine at San Diego State University, have been publishing results around adaptation strategies that coastal communities are using, particularly shellfish reliant industries and communities. And all of that work all along has been integrated in discussions with this oceanographic and ecological data. So as many of you who do really interdisciplinary work know, that sometimes we have to sort of cobble together, funding or resources or projects, but then we work on them and we think about them in an integrated space. And that's how we've been proceeding with this project. Esther and I will be thrilled to take questions in the time remaining. Emily hinted that I would do a little plug for the book. I think my publisher would not be very happy with me if I didn't take advantage of reminding you all or telling you all that we had a book come out last month. So this is a shameless plug here, but I will say it's not just a shameless plug for me. The work that we cover in this book includes 2 other projects supported by LenFest that are thinking about how climate change is impacting sustainable, resilient fisheries. So if you've been enjoying these LenFest webinars, I hope you'll consider picking up the book and checking out some stories in there about this exact topic, how people and communities are working together to keep those fisheries sustainable in the face of climate change. And with that, I guess I'll stop screen share for a little bit and we can take some questions.
Yeah, thank you both. That was great. And we have a couple of minutes for questions. And I see we have one in the Q&A, so we'll jump right to that. And other folks, please drop your questions in the Q&A and we'll see what we can get to. So this is a great question, actually, which is Sherry asked, this may already have been addressed, but how would a researcher go about working with the MoCA data sets? Sherry is interested in building remote sensing algorithms for seawater PCO2 and CO2 flux, and these data would be helpful to do so for the California current system. These types of algorithms exist elsewhere, such as Antarctica, and I would like to refine those types of algorithms for our region. Thank you.
I can take that. Thank you for the question. I think the easiest way to work with the data is, unfortunately, as Tessa alluded to, the data set is a little too big to work with unless you are familiar with a programming language. But if you are familiar with a programming language, it is very easy to download from NCEI, and Tessa just put the link to the data set in there. There's some very detailed metadata associated with that NCEI accession, and also if you in particular, if you use the programming language R, we have a bunch of code available on an archived GitHub that sort of helps you work with and get familiar with the data, so that you then just have an organized data set that you can sort of do with what you want. So I would recommend starting with NCEI. I would recommend looking at our archived GitHub, and if you feel so inclined, you can look at the paper too. But NCEI is going to be your first stop.
That is great. Thank you. And we have a question in the chat. Thanks, Glenn. Which fishing industry groups are you all working with? And are you keeping the Pacific Fishery Management Council up to speed on this research? And if not, he urges you to do so.
It's a great suggestion. I put in the chat. Thank you, Glenn. We have primarily been working with aquaculture groups throughout this, and so we would love to extend that to fisheries group, and we haven't connected with PFMC, but we would love to. So thank you for the suggestion.
Yeah, that's great. Also note in the chat, Maureen put in a comment about Senkus working on integrating the MoCA data set into their portals for analysis and visualization, should be online later this spring. And yeah, I would just put a plug in for Senkus, who has always been a great partner on management relevant science, and especially getting data available. That's really great. So thank you for that. We don't have any other questions at the moment. I'll hold it for a second in case anybody else wants to ask one, and just say, Tessa and Esther, is there anything, well, just in case somebody else drops the question, is there anything you want to say about the social science work that you weren't able to get to, maybe like a parting thought or anything like that?
Yeah, I'll do one parting thought. So there have been a couple papers that have already come out from our social scientist group, and they did interviews with aquaculture groups in Washington and Oregon. And one of the interesting things that shows up there is that ocean acidification is a worry and is something that people are working on adapting to, but it lands in a long list of worries. And so I think one of the things we need to be thinking about is sort of coming back to that first image I started with there's a lot of different change happening in the ocean, a lot of different adaptation that we're asking people to make. And providing the best information possible about that myriad of worry list is, I think, a high priority for all of us.
Great, thank you. And I know we need to wrap everything up. Oh, we just had a second one come in. Let's just see. Does your approach give us any information where future monitoring efforts will be most valuable? And this will be our last question. Kayla, if you can drop into the chat the contact information for all of us, people can follow up, and we will follow up with the additional papers when we send out the recording.
Yeah, that's a great question. And I think, yes, the work does elucidate some of that. So I would point to Esther's paper, which she summarized really well, definitely points out where data gaps are in our monitoring. And then Megan's work on species vulnerability, which is very close to being submitted, so hopefully it will be out pretty soon, actually takes that a step further and more deeply thinks about species of commercial economic importance and where we are monitoring during different life stages of those species, and fundamentally where we are not monitoring. And so those there are clear recommendations that are coming out of Megan's work around what we would need to do to gather the information that we need for these commercially valuable species.
I would just add on to that there's, I would say if someone's looking for a project, there's an open possibility of one of the things, as we mentioned, we did not really do with this project yet is make big interpolated maps. And so something that someone could do is, some monitoring stations are more valuable than others in terms of like, are they telling us new information, or are they telling us information that we could have just gathered from averaging across two stations we already have. And that is not work that we have done yet. But that is something that I in particular would urge somebody else to do because I would like to read it.
Cool, thank you. Thank you both so much. And we'll wrap up the webinar now. Thank you everyone who was able to join. And we will be sending the recording out along with all of the resources that went into the chat to all of you over the next couple days. So thank you so much for joining everyone. All right, have a good one.